Okay, so we're going to continue talking about different sources of DNA damage that occur in our body cells and try to appreciate where all these different sources come from and realize how frequently our cells' genomes are under sort of attack from being damaged by these different sources. So last time we looked at this slide and we were talking about mistakes that just happen during DNA replication. And we said that even though the replication of DNA, the functions of DNA polymerases and proofreading enzymes and mismatch repair enzymes are very effective at catching mistakes during replication, some mistakes do slip through. So that in itself tells you that just by a cell dividing, it's going to get certain mutations at certain nucleotides at random throughout the genome. So today we're going to look at these two other threats of mutation. So we'll start off talking about endogenous biochemical processes. So in addition to the DNA replicating and just making mistakes as it replicates, the DNA, of course, is within a living cell that has all kinds of metabolic processes going on, and that means there's all kinds of other molecules floating around that might be byproducts of things like metabolism that are potentially very reactive and can cause damage. This might result in things like nucleotides being improperly removed from the DNA, or nucleotides nucleotides actually being modified. So let's take a look at an example of that. So depurination has to do with the removal of a purine, so either a G or an A from a strand of DNA. So here you have a guanine, right? And here is the ribose sugar and the phosphate backbone. So this orange part on the left here, right, is the backbone of the DNA. And so here's the guanine sticking out. And of course, this would then react through hydrogen bonding from these 3 points, the oxygen, this hydrogen here, this hydrogen here, to go across the ladder to attach to a C on the other side. Right? So with this hanging out here like this, though, it's quite possible that you can actually lose the guanine. The guanine can fall off if there's a hydrolysis reaction, so a water molecule actually causes the nitrogen over here to just get a hydrogen. So as a result, the base just falls off, leaving this type of a situation on the right, what we would think of as a depurinated ribose, deoxyribose. Okay, so if this DNA strand were still part of a double helix, maybe this isn't such a big deal, because when mismatch repair enzymes or other types of DNA repair enzymes come along, they could still see that this missing base, this depurinated deoxyribose location, is across from something like a cytosine, right? And so therefore, it would know that the thing that needs to be replaced here is a guanine. But the problem happens if the two strands of DNA are being separated. For example, during replication, if this has now been separated from the other strand and is being read, for example, as a template strand to make a copy, if this depurination event happens, then when the polymerase gets to this point, it doesn't know what's supposed to go here on the copy strand. And so as a result, it needs to put something in there. And so it could be one of any of the 4 nucleotides that ends up being put in the copy. And so that obviously can result in mutations then that get passed on that way. So how frequently does this happen? It happens a lot. So it's estimated that about 10,000 purine bases are lost in one copy of the genome per cell every single day. Right? So by the end of today, a given cell in your body, especially if we're talking about a fully differentiated cell, will have lost 10,000 purine bases randomly across its genome just by you getting through another day of your life. Right? Another thing that can happen is you may end up with situations where you deaminate the nucleotide. So for example, so deaminate means we remove the amine group or an amine group. So in this case, you have a cytosine, right? And the amine group is removed from the top through some sort of accidental reaction. It's replaced by an oxygen. And so now the cytosine has actually become a uracil. An adenine, likewise, can go through a reaction where the amine group is removed and becomes hypoxanthine. Guanine can become xanthine. And 5-methylcytosine. So remember, this would be a cytosine. So compare it to the cytosine on the top here. The idea is if this were a cytosine in a CPG site, for example, in the promoter of a gene that gets methylated, right, this hydrogen position here, or this carbon position 5, is going to have this hydrogen replaced with a methyl group, right? And then we say that this is now 5-methylcytosine. So it's quite possible that this will spontaneously biochemically react so that it loses its amine group. Right? It deaminates, and if it becomes an oxygen here, then all of a sudden you've actually created a thymine instead. So that's why when we learned about CPG sites, we said that the actual number of CPG sites throughout your genome is far less than what you would expect by chance. And that's because over evolutionary history, places that have become even temporarily methylated like this, the cytosines get deaminated and become thymines. And then therefore, as the cell continues to divide, it's going to think that there's a thymine at that position, and that gets passed on to all daughter cells. So what types of molecules floating around can cause problems and damage to DNA? I mentioned that a lot of times it's metabolic byproducts. The fact that your cell is alive and it's undergoing catabolism, breaking down different types of larger macromolecules that might be nutrients from the food that you're eating, means there's going to be a lot of intermediate molecules, things that are not really end product molecules, but are sort of halfway being processed and therefore are potentially very reactive. So many of these are what we call reactive oxidation species, or ROS. So in that case, these are molecules floating around that have a tendency to oxidize other molecules they bump into. So here we have a deoxyguanosine, right? So again, you can pretend this is in your DNA. So here, conveniently, they just label this as the deoxyribose sugar would attach here. So you imagine there's the deoxyribose sugar, and then the phosphate backbone is probably over here somewhere, right? So this particular nucleotide, again, these three locations, this O, this H, and this H are what are going to form the hydrogen bonds across the DNA to the other strand. So if this gets oxidized by one of these reactive oxidation species coming up and oxidizing it here, you can look at this and you might think, well, I don't see what the big deal is, because if these are the parts on the left here that form the proper bridge across from this G to the C on the other side, then who really cares if you have an oxygen on this side? It matters because if you think about it, the positioning of this base, in other words, its position in three-dimensional space sticking out from the phosphate backbone, is dependent upon it being this exact shape. If you suddenly stick an oxygen to the end of it, you can imagine this thing will not be sticking out at the same angle anymore from the phosphate backbone as it should be. And as a result, the hydrogen bonding across these three sites won't be as encouraged to properly connect with the C on the other side of the, on the other strand. As a result, because of that sort of tweaked angle or misalignment, you can actually cause the G to improperly form hydrogen bonds with something like an A across on the other strand, right? Simply because the oxygen that was added onto it by this reactive oxidation species that floated by, right, causes this now to be tweaked at the wrong angle. And so you will get a mispairing, right? And so when then this mispairs, you can imagine the shape of the double helix won't be correct. When a enzyme comes along and detects that there's something wrong and needs to fix it, the tendency will actually be fit to look at this side as being the normal nucleotide and this side as being something weird. So this side might be removed, the G might be removed, and what gets put in place of it as part of the repair would be whatever is supposed to complement the A, right, which would be a T. So then you've actually caused this G to become a T in this strand of DNA. So how do we fight against things like these ROS or these reactive oxidation species? Because obviously there's tons of these ROS-like molecules floating around in your cell all the time, right? Because your cell is constantly breaking things down. So there's going to be all these intermediate halfway processed molecules that want to react. We have a very important molecule known as glutathione in our body that helps protect us against ROS damage. So glutathione is really interesting, right? So it's often referred to as GSH. And you can see the makeup of glutathione is actually 3 amino acids put together. You have glutamate, cysteine, and glycine linked together to form this one large kind of composite amino acid, if you want to think about it that way, right? So this is a neat example, again, of reminding ourselves that many of these basic building blocks of things like amino acids are used in different ways in our body. Right? Because over evolution, we've come up with useful other variations of them. So by linking these 3 together, you form this GSH molecule. And here's the sulfhydryl group that sticks off the cysteine. Remember, this is the sulfhydryl group that helps cysteine form disulfide bridges with other cysteines, so that when the finished protein is folding up, oftentimes these disulfide bridges from one cysteine on one part of the protein interacting with a cysteine from another part contributes to the overall shape of the protein. So these disulfide bonds can be very strong. So this glutathione then has this sulfhydryl group. So here's the GSH, the glutathione. Here is an example of a reactive oxidation species. So this is just some sort of generic type of intermediate molecule that came from metabolism. It has this oxygen group on it, which, because of its positioning, very easily could end up reacting with something else that this molecule bumps into. Maybe, for example, a base in the DNA, just like we saw on the previous slide. And so what happens is this SH from the glutathione can interact with this and help neutralize this. How? It is helped by an enzyme called glutathione S transferase, or GST. So this is part of the GST structure here. And the idea is that this enzyme can, at the same time, bind to GSH as it also binds to some other reactive oxidation species, like this one here. And so the function of this GST enzyme then is to essentially transfer the sulfhydryl group onto this particular carbon of this molecule. Right? So this is essentially the whole glutathione now dangling off the end here. And then another molecule of GST can come in and actually remove the glutathione, turning that into then 2 hydroxyl groups, which is far less reactive than the original oxygen that was just kind of hanging on like that. Okay. So GST, this GST enzyme then, is a very important enzyme for inhibiting or limiting the amount of reactive oxidation damage that our cells experience. So it's not surprising then that levels of GST in a cell can actually have an influence on how much mutations a cell undergoes. So oftentimes during tumor formation, the GST gene is accidentally turned off by mutations. And so without the help of GST to be able to transfer glutathione to neutralize reactive oxidant species, you can imagine then that such cells that have lost their GST expression incur or suffer from a much higher rate of metabolism-related mutations. So in this particular slide here, we're looking at a section of prostate epithelium. And what we are looking for here is the levels of GST. So the cells that have normal levels of GST stain very dark. So these black ones here are the epithelial cells that would have normal levels of GST, protecting them from metabolic damage. to their DNA. But you can see these cells here that are not fully turned into cancer yet, but they are transforming. So we still consider these a neoplasia, right? They're beginning to proliferate at a higher rate, but they still sort of have normal morphology. So in this case, this is prostatic intraepithelial neoplasia, or PIN. These PIN lesions are early signs of the development of prostate cancer. And you can see that they no longer stain very black from GST. Their levels of GST have gone down dramatically. And so that might suggest that they are getting more and more damage from metabolic processes damaging DNA. So when we think about the burden of DNA damage coming from things that are emanating from our metabolism, you can't help but think about, well, the food that we're eating, as we're breaking that down, and different types of nutrients are being absorbed by cells and utilized by cells. How many of those types of molecules that are coming in from our food and being processed by the metabolism and turned into intermediate molecules actually cause damage? These days, of course, we're very sensitive to this idea of things like preservatives or artificial types of food additives, especially things like industrial pesticides and things like that. We're very worried about how those things damage us. But how big of a threat is that really compared to just the number or the amount of naturally occurring xenobiotic molecules that are just in food? So our daily food consumption contains anywhere from 5,000 to 10,000 different types of natural compounds in the foods that we eat. In our modern diet, about 2,000 milligrams, or in other words, 2 grams of that, is burnt material, just because we cook our food now. Right? And so that burnt material certainly can have reactive different types of molecules in it to damage our DNA. About 1,500 milligrams, or about 1.5 grams, are actually naturally occurring pesticides. So products that are made naturally by plants to protect themselves against pests, like bugs. And so not surprisingly, the way that many of these natural pesticides work is that they interfere with normal cellular function, and that's why they are toxic or poisonous to bugs. But they can also have effects on us as well. So for example, you think about for coffee drinkers or tea drinkers, caffeine is a great example of that. Caffeine is a naturally occurring pesticide. Plants make caffeine, not because plants want to have a nice buzz, right? But the whole point is that when insects eat the caffeine, it actually frazzles their nerves, right? And that can actually be toxic. So for us, of course, a little bit of caffeine is not going to kill us, but the whole buzz you get from the caffeine is a reminder that you're ingesting a naturally occurring pesticide. So the average daily exposure, though, to synthetic pesticides, the things that we worry about when we read about Roundup and all these other things, is surprisingly low compared to the burden of natural pesticides that we consume. We only consume typically about 0.1 milligrams, right? So less than 1,000th of the compounds are actually from artificial pesticides. The majority of things that we eat are naturally occurring chemicals that can damage our DNA. So this is an interesting to think about, because if we say that burnt material from cooking is a big contributor to this, if we think about when human beings actually started cooking food, so it's estimated that was about 400,000 years ago. So prior to that, for millions and millions and millions of years, there were no other mammals that actually cooked food. So that also implies then that our body, the evolution of our body, in terms of what our cells are expecting, in terms of a burden of burnt products, right, is far lower than what we are actually now putting into our body as a result of cooking. So you might ask, well, what impact does this have on our cancer burden? It means we are now exposing our bodies to much higher levels of burnt material than evolution actually intended us to have to deal with. So, you can say, is cooking food another example of human behavior being too fast, too soon, compared to what evolution, the pace of evolution? So I think the clear argument, though, is that there are many benefits to cooking food. For example, killing bacteria, destroying other pathogens, allowing food to keep longer, that balance out, right, the burden of additional cancerous mutagens that we're eating from the food. So at the end of the day, it's an interesting kind of argument to think about. So one of the important players in repairing DNA damage when you have double-stranded DNA breaks is BRCA1. And so BRCA1, as we've learned about, is one of the genes that's controlled by P53. So when P53 halts the cell cycle in response to DNA damage, it wakes up genes such as BRCA1. And so how important is BRCA1 in repairing DNA damage? It's very important, because if you look at the presence of BRCA1 or BRCA2 germline mutations in inherited cancers, so familiar cancers, you'll find that over 50% of cases of familial breast cancers, the individual has evidence of BRCA1 mutations inherited as part of the germline DNA. Over 70% of familial ovarian cancers, you will find evidence that there are BRCA1 or 2 mutations. Right? Sporadic mutations, in other words, people who are born with wild-type BRCA1 without any kind of compromising allele, and they might get sporadic mutations later. So their BRCA1 gene locus gets a hit in one of their cells just by bad luck during their life. That type of mutation accounts for over 20% of breast and ovarian cases. So this figure down here just shows a staining of where BRCA1 in green is hanging out inside of the nuclei of 2 cells. So this is under normal conditions. The BRCA1 is kind of scattered around inside the nucleus. Keep in mind that the whole genome, the DNA, is all unwound in here as well. And so this is normally if the cell is left alone in the tissue culture and you don't treat it with anything that might damage the DNA. So minus this hydroxyurea compound. If you add the hydroxyurea, that causes double-stranded breaks in the DNA. And you can see what happens is the BRCA1 proteins begin to localize because they start concentrating at spots within the genome where their action is needed to help repair the breaks. Over here in red, these cells are stained for a protein known as PCNA. So PCNA is a protein that's involved in helping DNA polymerase function. So you can imagine that if you have damaged DNA and repairs are being made, you're going to need to have DNA polymerase replacing or making new strands or new sections of DNA. And so the PCNA is helping that out. So you can see the PCNA also localizes to regions where there is DNA damage. And if you overlap the BRCA1 and PCNA staining, so the sort of yellow or white is where these 2 proteins are tightly working together, you can really appreciate then how these different pathways have to come together to properly respond to DNA damage. If you lose BRCA1, this is what happens to your genomes. If double-stranded DNA breaks are not repaired, then you have all kinds of problems with the loose ends of DNA of chromosomes linking up improperly. So you can see very clearly here in this image, you have some examples of chromatids that might somewhat appear normal, right? So these are individual chromatids or condensed chromosomes here, but you can see some of them have broken and are now joining up improperly. So you get all kinds of pieces that come together and these strange fusions, which clearly is not compatible for the cell to function properly.